Hey everyone, welcome back to Chapter 4 of Genetics. Today what we're going to talk about is allelic interactions. And that's basically the biggest part of this chapter is talking about how alleles interact with one another, but also how different genes also interact with one another as well, which we're going to cover on later in the chapter as well as we go through it. But to begin with, we have a couple of things to talk about where we've sort of already mentioned complete dominancy, essentially where you have one dominant allele and then you have one recessive allele. And then essentially this one dominant allele is going to be expressed over the recessive allele. And when you have a genotype such as big A, little a, you're going to have that dominant phenotype, whatever that big A encodes for. So we've already sort of talked about complete dominancy. Incomplete dominancy, on the other hand, is essentially where you have two different alleles that encode for a specific product, and then this specific product is blended together to create a certain phenotype. So yes, so you have two alleles that produce an individual phenotype, and when you combine these alleles together in a heterozygous state, you end up with a blend of phenotypes. So basically what this means is that let's say we had a red flower, and then we also had a blue flower. If we cross these together, we should get a flower that is a blend between these. So let's say, I actually don't know what the blend between these are, but let's say it's pink. So this would be a case where this is called incomplete dominancy. And then we can use that same analogy over here. We can say the big A for complete dominancy is red phenotype, and then let me rewrite that. And then the little a is white phenotype. So what's going to happen here is that we have a white flower, we have a red flower, and then when you cross these together, you end up with a red one. Right? And then codominancy is another one that we have to go over. So this one is essentially kind of the same thing as incomplete dominancy. But the thing is with this one is that instead of blending the two phenotypes together, you're actually combining those phenotypes into a single one. So what that means is, let's say we had this red flower and we crossed it with a blue flower. In a codominant situation, we should get a flower that is red and blue. So something like this, it has red petals or whatever they're called, and there's some red ones as well, some blue ones. So that'll be something like codominancy. So basically, two alleles that produce a phenotype, and then when you combine these together, you end up with an individual that has both phenotypes at the same time. A good example that you can go over for these is the ones in your textbook where essentially it's talking about human blood types. So for example, AB blood type is a codominant situation where you're producing both A antigens and B antigens on your red blood cells. Yeah, there's no incomplete dominancy for blood type. And then for complete dominancy, it's A is complete dominant to O, and then B is also complete dominant to O as well. So you can refer to that part on your textbook for some more, practice on that. So hopefully that made sense. Another thing that another critical thinking step that you have to think about is, we're getting these this recessive mutation right here, or this white individual that is, that has the phenotype that is white, but how is it that they're white? So the reason why most of these, and the reason why you have a lot of complete dominancy situations is because the recessive allele typically has a null mutation or a hypomorphic mutation. So what these terms mean is that, like null right here, that means 0. So what that means is, let's say we had a enzymatic graph of whatever gene we're studying. So let's say on the y-axis we have activity, and on the bottom we have our mutation, so the null. So the null mutation will essentially look like, and let's have a positive control here as well. So this is going to be positive control. So yeah, that'll be the positive control, big A. So big A will have the full activity level, but then the null mutation basically has no activity at all. Okay, so it'll look something like that. So hopefully that makes sense. And then the hypomorphic one is essentially similar, but you would have to use this keyword hypo, which means less. So on the y-axis we have the same thing, and then this is our positive control on the right, big A. So our hypomorphic mutation is something that has a lot less activity than our big A allele. So that'll be a hypomorphic mutation. So most of these recessive alleles are either null mutations or hypomorphic mutations. But how do we get a recessive allele? So either there are two possible possibilities that you can have. Either you mutate the gene, such that the mutation affects the active site of the enzyme causing a null or hypomorphic mutation. So that's the first one. And then you have a second one, which a lot of people don't really think about, is that every gene has something called a promoter. So let's say this is our gene right here. Every gene in front of the every gene downstream of the gene, you have something called a promoter region. And for now, what you can think of the promoter region as something that it's a region that basically turns on or turns off transcription of the gene. So if you mess up the promoter region by a mutation, then therefore you also inhibit the production of the gene. So mutation in the promoter region. Affecting enzymatic concentration. So yes, so the second one, for example, let's say you mutate the promoter of this gene right here to the point where you're not making any gene at all, and that will contribute to you having a null mutation or a hypomorphic mutation. So hopefully that made sense. And then there's two more to take into account. Hypermorphic is, hyper means more. So if we have the same graph, so this one will be something like this. So hopefully that makes sense. And then the neomorphic one is essentially, the way how you can think about this one is the neo means new. So this new mutation, or this mutation causes new functionality of the protein. And that's basically it for that. It's pretty simple. But now the thing is, let's see if we can actually figure out how to interpret data for us to determine if a situation is if a trait has incomplete dominancy or codominancy, such as something like these problems right here. So to begin, before we do anything with that, I just want you to remember something in terms of independent assortment and how you cross these individuals together. So if we had a complete dominancy case like this, you would have a 3/4 big A whatever, and then 1/4 little a ratio, correct, for a phenotype. And then for the genotype, you would have basically a 1/4, 1/4, 2/4, 1/4 genotypic ratio, where this is big A, big A, big A, little a, little a, little a. Now, but for incomplete dominancy and codominancy, the heterozygous individual, big A, little a, has a completely different phenotype from the other alleles. So for example, let's say that big A is blue, and then little a is red. If you're big A, little a, then therefore you're in an incomplete dominancy case, you will be big A, little a. So basically, what that means is that every single genotype has its own phenotype for both incomplete and codominancy. So what that means is that in a case where you have to figure out the products of a certain cross together that has incomplete and codominancy, you would actually have to use the genotypic ratio. So basically, the genotypic ratio is equal to the phenotypic ratio, because these guys are all different phenotypes. Okay? So in a case where you're getting some data set, where you get a data set and you're getting a situation where you have 1 to 2 to 1 ratio, that will mean that you have some sort of an incomplete or codominancy, and you also have to take a look at the phenotype descriptions as well. So hopefully that kind of made sense. Let's do a practice problem over here. I haven't calculated the totals yet. Let me take some time to do that. So this total right here is 4, 6, 8, 7. Okay. So because of what we're going to do now is we're going to look at the phenotype descriptions. So what we have here is essentially purple flowers you're crossing with blue flowers, And then their F1 had all flowers that were purple and blue. And then when you cross these together again, you're getting an F2 data, which is you have some purple, some blue, and some purple and blue. So just by looking at the phenotype descriptions, you can already see that there's a different phenotype right here, which has purple and blue, which is a hint that this is codominancy. But we also have to confirm that it is in fact codominancy, okay? So the way how we can confirm that is by finding the ratios of this data set. So the way how you would do that is, if you remember, you would take the number of the phenotype, you would divide it by the total, and then you would multiply it by the denominator of how many of a monohybrid cross, because we assume that there's a single gene acting on this population. So yes, so let's do that. To get purple, it's essentially multiply by 4. So this 4, the reason why we're multiplying it by 4 is because if you remember from previous videos, if you have watched it, the 4 is essentially the denominator of a monohybrid cross. And the reason why we're trying to find a ratio is because unlike a proportion, a ratio gives you a bigger picture in a case where it's a lot more complicated than finding just a proportion, because a proportion doesn't really tell you anything about how these genes are independent, or how these genes are being crossed over. And basically, the ratios will make a lot more sense in other problem where it's a bit more complicated. But for now, we're going to use this essentially right here. Blue, we're going to do 1174 4687, and then so the combined is 2353 4687. Let me take some time to figure these out. So this is around 1. This is around 1 as well. And then this is around 2. So what we're getting here is we're getting a 2 to 1 to 1 ratio, or basically a 1 to 2 to 1 ratio. So basically, this is a single gene and there is a codominant allelic interaction. Causing the F2 to have a mix phenotype, and it's also in a 1 to 2 to 1 ratio. So that one was a pretty easy one, but let's do a little bit more of a complicated one where you have something like this, and this is where students sort of get confused on how to do it, because it looks very complicated. There's a lot of different phenotypes to take into account, but I promise if you understand how to do the other problems that I've showed so far, you would understand how to do this problem very easily as well. So let me take some time and figure out what the total is real quick. So the total over here is 737. Okay, so what you should do now is you should read the problem. So what's happening here is you're crossing a lab rat that has normal activity levels of glycolysis, and you're crossing it with a lab rat that has no glycolysis activity levels. And then it tells you before in the problem, it tells you that hexokinase and phosphofructokinase are important enzymes. So then when you read the problem, it tells you next is that the F1 were all lab rats that had lower activities of hexokinase, but normal activities of phosphofructokinase. So then you cross these together and you get a data set that is like this. So if you remember from my previous videos, you should always separate out the traits into their own separate calculation, right? Because right here what we have is we have a very complicated situation where we have two different traits, and these two different traits, which are just hexokinase activity versus phosphofructokinase activity, we need to separate those out so we can get a better picture of what's happening over here. Okay, so first of all, when this is referred to normal glycolysis activity, that means both hexokinase and PFK are fully active. Okay, and then this one means the opposite, no activity for both. Okay, so what you should do is we should pick a trait that we want to focus on, and we should do it one at a time. So let's say we're going to ignore the PFK trait for now. So we're going to cross those out. This one as well. And we're going to ignore it for now, okay? So what we have here is that we're gonna combine all the individuals that have normal hexokinase activity, combine all the individuals that have slightly lower hexokinase activity, and combine all the individuals that have no hexokinase activity. Okay. So what we have here is normals are right here, 140 plus 42, which is 185. So 185 have normal hexokinase. So then you having slightly lower, it's 270 plus 94. It's 364. We're going to call that lower. And then 145 plus 46. 191. No activity. Okay, does that kind of make sense? And so now what we're going to do, same thing, we're going to figure out the ratios for this, and we're going to use the total that we have over here. So to figure this out, it's essentially 185 divided by 737. We're going to multiply by that by 4. So we get a ratio that is equal to 1 over here. Next, we're going to do the exact same thing with 364. We get a ratio that is equal or closer to 2. And then 191, we get a ratio that is closer to 1. So essentially what this means is that the levels of hexokinase, or the activity of hexokinase, is dependent on an allelic interaction, which is incomplete dominancy, because we have a situation where the individual that is big A or little a has a blend of activity. Either you have a lot of activity or you have no activity. So the genotype in the middle has sort of a mid-range of activity. So hopefully that made sense. And that's basically how you do that problem. We can also try doing it on, we can also try the other one, PFK, as well. See how that one is also interacting. So same thing, what we're going to do here is we're going to ignore the hexokinase activity. But now we're going to take into account that PFK, you either have activity or not. So PFK over here. So the ones that have activity are these ones right here. This one has activity, this one, and then this one. So essentially 140 plus 145 plus 270. You have 555 individuals that have full activity. And then in order for you not to be active, it's 42 plus 46, and then plus 94, 184, no activity. So we're going to do the exact same equation right here. We're going to do 555 divided by the total, and we're going to multiply it by the denominator of a monohybrid cross, which is 4. We get a ratio that is 3 over there, and then 184, 3-to-1 ratio. So basically that means that PFK, you're getting the expected 3-to-1 ratio, and PFK is essentially just complete dominancy. Also, you can also say that the genes are independently assorting as well, but that's basically how you do that problem. So hopefully this kind of helped out with essentially looking at how to solve these problems, and yeah, that's basically it. Hopefully this helped. Have a good one.